The overall goal of this procedure is to isolate subsets of precursor B cells from umbilical cord blood. This is accomplished by first isolating mononuclear cells from cord blood by density gradient centrifugation. In the second step, cell surface antigens are labeled with biotin conjugated antibodies to magnetically deplete any of the remaining non-B cells. Next, the isolated B cells are fluorescently labeled with antibodies against the CD19, CD34, and CD45 cell surface antigens. In the final step, the cells are sorted by flow cytometry to recover the precursor B cell subsets. Ultimately, cells of sufficient number and quality can be acquired for utilization in downstream assays requiring high-quality DNA and RNA.
One advantage of this cell sorting strategy over alternative methods is that our strategy requires less time on the flow cytometer and only 3 fluorescent antibodies, greatly reducing the cost of the experiment.
Generally, individuals new to this method will struggle with the variety of methods available for preparing umbilical cord blood cells for flow cytometry and the complexity of the preparation of the flow cytometer.
Though this method can provide insight into healthy precursor B cells, it can also be applied to other cell types present in umbilical cord blood, such as T cells, or to studies of diseases involving any hematopoietic cell, such as leukemia or lymphoma.
Visual demonstration of this method is critical because it is difficult to describe the proper techniques